Good afternoon, everybody. We're supposed to start promptly at 5:30. It turns out my colleague, Dennis, will be delayed for maybe 5 more minutes or so, we hope. But I sort of dedicated to starting the meetings on time. Let me go ahead and see if we have anything of a bureaucratic nature we need to take care of before we go. The agenda says we're going to hear about the Elk Hoof Disease, and I'm pretty enthusiastic over that because that's been a problem fairly evident around here for a long time, and research into it was always hard to find. And about 4 years ago or so, is that right, Dr. Wild? 5 years ago now, we met in that room, and I was pretty enthusiastic that a real scientist was going to go and take a look at this, and we discussed some things. That looks dangerous. And so I look forward to that. I'm trying to stall a little. Okay. Thank you. Rick Dahl is on vacation, so it'll just be the two of us. And I'm happy to introduce Dr. Wild, who is known to some of you. She is Dr. Margaret Wild, a DVM and a PhD, which I think is a good thing. In fact, it's a doubly good thing. I don't have a full bio on that, but I would like her to give us essentially what's happened in the 5 years, and is there, well, you can take over from now if you will.
Well, thank you very much. I really appreciate the opportunity to be here and talk with you guys today. What I thought I'd do is give you about a summary of the work that we've been doing the last 5 years. Just to start off, a little bit about me. I'm a professor in the Department of Veterinary Microbiology and Pathology in the College of Veterinary Medicine at Washington State University, and I've worked with wildlife disease my entire career, primarily with another disease of elk and deer called chronic wasting disease, but came here to WSU 5 years ago to begin this program in elk hoof disease. And let's see, do you want me to pull this closer and move myself? Tell you? Okay, next, please. So most of you guys are very familiar with the disease, and unfortunately, you probably see it quite a bit. But just to make sure that we all start on the same page, we're talking about elk hoof disease, also colloquially or locally often called hoof rot, or treponeme-associated hoof disease, or TAD for short. And I'll call it either elk hoof disease or TAD or treponeme-associated hoof disease kind of interchangeably. But this disease really started showing itself in the early 2000s and in this general area of Washington. And folks started reporting these elk that were lame, debilitated, appeared to be dying more frequently than other elk. And so Washington Department of Fish and Wildlife did some investigations and found these rather unique hoof lesions, which is a disease that hadn't been really identified before. And like I said, most of you guys have probably seen this, but this is a normal elk hoof. And when they get the disease, they can get very severe lesions. This is a preserved hoof of an elk that had a severe lesion. One of the claws is overgrown and the other one has actually sloughed off, leaving the bone underneath just a little bit of tissue there. And so you can see how that could really cause debilitation in animals. Next slide, please. I do like to point out, though, when we're doing surveillance and when we're looking for lesions, this is the most severe lesion, but before they get to this point, there's more subtle lesions. And it's important that when we're doing surveillance, we remember to look between the toes of those animals. And the earliest lesions are just little red sores, oftentimes right between the toes. And they get bigger and bigger and start underrunning that hoof wall until it can fall off or break off. So these are the kinds of lesions that we're looking at. Next, please. So a lot of the folks, particularly in this part of the state, again, were really concerned about this disease, went to the Washington State Legislature, and they directed Washington State University to take the lead on researching this Elk Hoof disease problem. And that was in 2017. I came to WSU in 2018 to start that program. It's important to note that although WSU is the lead on research, Washington Department of Fish and Wildlife, or WDFW, retains the management authority. So we're here to do research and kind of serve as a council and bring some rigorous science and recommendations forward. And we work collaboratively with the Washington Department of Fish and Wildlife, as well as other state departments of fish and wildlife and tribes, tribal wildlife managers as well, and other researchers. And we're very fortunate to have dedicated funding from the state of Washington to conduct this research. Next, please. My objectives, even though we're at a academic university, is not to do basic research. It's to do applied research. So everything that I want to be able to increase our knowledge base and help the managers make those decisions that they have to make in managing elk, and look for mitigating factors, ways we can try to reduce the harm from this disease. It's also really important to me to keep people that are interested in elk. I'll call anyone who's interested in elk a stakeholder. Keep stakeholders involved, aware of what we're doing, and ask for information. You know, what are folks seeing? How can that inform our research direction as well? Next, please. And when I was putting together the presentation, I thought, well, one way we could do this is, I'm a scientist, so generally we start off with a hypothesis, and then we have the methods and the results and the conclusions. And I thought, well, that's kind of boring. So maybe another way to do this is to take the questions that I get asked fairly frequently and bring our research in to answer those questions. So what have we done over the last 5 years that can help us answer these common questions? And the first one, next, is, have you solved it yet? And next, please. The short answer is no. And that is not unexpected in any way. Wildlife diseases are exceedingly complex. Animal and human diseases that are new emerging diseases that haven't been described before are very complex. But when you add wildlife on top of it, the fact these are free-ranging animals, they don't show up for appointments, we don't know their health history, we don't know what's happened to them over time, it makes these diseases very challenging to understand and to manage. And I think about it is like putting together a puzzle. And we've got to do that foundational work of getting a few pieces of the puzzle together first. And we're just going to continue to build on that over time. And when I started thinking about this, one of the first foundational things that I wanted to do was to kind of put a border on that puzzle and to understand, is the disease spreading either to animals other than elk or geographically spreading? And so that's where we started. First of all, with as far as other species, in wildlife, we've only confirmed the disease in elk. And we've gotten hooves from other animals, particularly some deer, some pronghorn, probably some other wild animal I'm not thinking of. And we've looked at those in the laboratory, and those were caused by things other than this elk hoof disease. It's not to say that the disease couldn't happen in those other animals, so we're continuing to conduct surveillance, but so far we have only diagnosed the disease in wild animals in elk. The next question is it geographically spreading? And you guys know that it's been apparent since the early 2000s in southwestern Washington. It was identified in a few other places also in that following about decade in western Washington and northern Oregon. And then we started doing some enhanced surveillance and working with the wildlife managers in Idaho and in California, and also asking the wildlife managers in Washington and Oregon to submit more hooves to us. And when we did that, we found the disease over a broader geographic area than we initially realized. And on this map, the red pluses are locations where we have confirmed cases. So those are cases that actually came to the lab and we did the laboratory work and confirmed them. The pluses aren't every case, it's just locations where we have cases. The black circles are areas where we've gotten a case and looked at it in the lab and it turned out to be something else. And I kind of feel like, particularly over there in Idaho, we've got a number of negatives. And so we don't know that the disease doesn't occur over a broader area, but I feel like we got a pretty good idea of this is its current distribution as far as we know. And again, it's a much larger area than we initially realized. Folks here in Washington are most interested in Washington. So looking at a map just of Washington, the way I like to look at disease spread is on the finest scale that we can that will allow us to see changes over time. And this is a map that is broken down by game management units or GMUs. And those are delineations that wildlife management agencies put on the land for managing the wildlife. And these GMUs that are in orange are ones where we have confirmed cases, where we've taken hooves into the lab and confirmed the cases. In blue, managers and stakeholders have seen a lot of, or at least have seen a number of limping elk, and we have not gotten hooves from those animals to confirm the disease in that area yet. So I'd call them a suspect GMU. And those are areas where I'd really like to continue to get elk hooves, abnormal elk hooves from. So if the disease is for certain occurring there, we can fill in this map further. But you can see the disease is primarily on the western side, but we have detected it in some areas east of the Cascades as well. Next, please. So the next question is it really caused by bacteria? And when I came in, this was a pretty contentious issue. Next, please. Washington Department of Fish and Wildlife, along with some collaborators, had just started showing the association of these lesions with a certain type of spiral-shaped bacteria called Treponema bacteria. And because of that, the disease was given the name treponeme-associated hoof disease. It's important to point out, even though these treponeme bacteria are associated with the lesions, the cause of the disease is still not known. And the cause or even the association and these treponeme being there as primary lesions, not just like a secondary invader to something else like herbicide or fertilizer exposure or mineral deficiency, was pretty contentious. So one of the first things we wanted to look into was there additional evidence for the involvement of these bacteria? And in our research that we did where we were doing the surveillance and documenting the disease over in the maps that I showed you, we also did a kind of DNA sequencing where we can identify the DNA in a sample that comes from a lesion and tell what type of bacteria is there. And what we found was additional evidence that definitely the treponeme type bacteria are hallmarks in these lesions. However, there's 3 other types of bacteria that we consistently saw in the lesions as well. And those were the Mycoplasma, the Porphyromonas, and Fusobacterium. And interestingly, this group of bacteria is similar to a disease in cattle called bovine digital dermatitis or hairy heel wart. And those same 4 bacteria are the ones that are most frequently associated with bovine digital dermatitis as well. So we don't know if this is the same disease, but the same types of bacteria. We don't know if they're the exact same bacteria, but the same families of bacteria are present in both types of these foot diseases. Next, please. To learn more about these bacteria and to really look into whether this is in fact a transmissible infectious disease, that is primarily a disease that is transmitted from one elk to another. We worked in a captive research facility, and this is a cornerstone of the research. This is some of my background. is working with wild animals in captivity to learn more about their diseases, where we can control the variables in a way that we can't do with free-ranging animals. And so we built a research facility, a biosecure research facility in Pullman on the Washington State University campus. And we keep elk in pastures. And then when we're doing experiments, we have 10 biosecure concrete lined pens where we can do the research. Next, please. And so one of the first experiments that we did was with a master's student named Zach Robinson. And Zach actually grew up in Longview and was very interested in elk hoof disease. And so he came to WSU, and we did this study where we looked at the transmission of elk hoof disease using captive elk. We had 4 elk that were kept in pens. They had a portion of their pen had dirt that was kept moist. And we chopped up hooves that looked like this one and put them into the mud. And then we also took bits of the hoof that were chopped up, mixed them with soil, and put them right up on the foot of the elk. And if you look real close, you can see that on 2 of that elk's feet, he's got little bandages, and those are just holding the inoculum, the chopped up hoof and the soil up against the foot to increase the contact time. And what we found was after about 4 months, we had persistent lesions in all 4 of our elk. And they visually looked just like tad in free-ranging elk. When we looked at them under the microscope, they looked the same as tad in free-ranging elk. And when we took bits of the tissue and did the DNA analysis, we found those same types of bacteria with the treponemes, as well as some of the other bacteria that we see in free-ranging elk. So we had reproduced the disease. It did take a little while. It took, we started seeing lesions within about 2 months, but it took 4 months after we started the challenges. And we had 8 challenges that we conducted, exposed the elk 8 times to this ground up hoof material. But by 4 months, they all had clear and persistent tad lesions. By 5.5 months, 3 out of the 4 elk needed to be euthanized. All 4 of them had pretty severe lesions. They hadn't gotten to this point. For humane reasons, we don't let them get to the point where they actually slough off that hoof. But they had moderately severe lesions. And so 3 of them were euthanized. Interestingly, 1 of them recovered. We don't know why that one recovered. His lesions were just as severe as the other elk. There was clearly something different about him. But at this point, we don't understand what was different about him. What it did show, though, is animals can recover from the disease. However, I think it's really important not to rely on animals recovering in the wild as a fix for this disease, because these guys lived in pens. Somebody brought them food every day. They didn't have to worry about getting away from predators or from cars or from people. And so their life was pretty easy. And they can recover, but let's not rely on it. What this study showed us is a strong evidence that there's not just an association with these bacteria, but these bacteria are causal, and that this is a transmissible infectious disease, so transmitted from one elk to another. And the reason that's important is because the way managers address a disease is going to be different, whether it's a transmissible disease or say it's from a point source of contamination, like from herbicides or from a mineral deficiency. If that was the case, and that being, if it was from a point source, like a mineral deficiency or herbicide exposure, if you removed that one animal, that wouldn't help the population at all. However, if you have an animal that is infectious and spreading the disease to others, if that animal is removed from the population, then that's one way you can break the chain of transmission and perhaps get control of the disease, or at least make sure that that one animal isn't continuing to spread the disease to others in a group. So that's why it's important to know that it's a transmissible infectious disease. Next, please. And knowing that there's bacteria involved in this, another question that often comes up is, does it affect the meat or the antlers? What we specifically looked at is the meat of the animals that we conducted this research on. And we looked at the animals that had these moderately severe lesions and didn't see any problems with the meat or with our eyes. We didn't see any problems with the things like the liver or the heart, things like that. Underneath the microscope, there was some really low level inflammation, but nothing that would have made us think that that wouldn't have been fine meat to eat. Now, that said, what's important to remember is, remember I said we euthanized those animals before they got really debilitated for humane standards. Elk in the wild probably get much more debilitated and can be down, can have secondary infections. So because of that, it's really important to use good hygiene and good common sense when evaluating the meat of an animal that's harvested. But what it showed is that just because they have hoof lesions doesn't mean that automatically the meat is bad. And I just realized one thing that I forgot to say from an experimental design standpoint along this, both the meat of the treatment animals, so that the elk that were exposed to chopped up hooves that look like this, and we also had 2 other elk that were treated completely the same way as those 4 elk, but they got exposed to chopped up elk hooves from a hoof that looked like this, so normal hooves. And so they were our control animals. And those animals stayed healthy throughout the study period. They never had any lesions, nothing visual, nothing microscopic, and nothing as far as when we looked at the DNA of the bacteria for those bad bacteria, they did not have them. So that was a good comparison to know that everything else the same. The only thing different was the treatment elk were exposed to the hooves with hoof disease already. So that's kind of the summary of the transmission study. The next one is a little bit different. And this question actually came up when I was over here visiting several years ago. I think it was when I was visiting, it was about four or 5 years ago. And a lot of folks told me that there was antler asymmetry, or these odd looking antlers in elk that had hoof disease. And so I started thinking about that and thought, well, we don't want to put a lot of emphasis into this, but elk antlers are important to people that view and hunt elk. They're also important for elk for their hierarchy and breeding. And so I thought, well, if we could get at this question relatively easily, let's do it. And fortunately, Washington Department of Fish and Wildlife gets reports from hunters every year. And that information includes the number of points on the left side and on the right side, what game management unit the elk came from, and in the last several years, does that elk have the presence of any hoof abnormalities? And so we had hoof abnormalities, yes or no, and we compared the 2 groups and looked to see if they were similar, the 2 groups were similar as far as that asymmetry in the antlers. What we found was in support of what people had observed, and that definitely was that there's a strong association between antler asymmetry with hoof abnormalities. And if an elk had hoof abnormalities, it had double the odds of having asymmetrical antlers. And as the bull got bigger, from 4 points to 5 points to 6 points, they were even more likely to have asymmetrical antlers. So that was a very interesting kind of stakeholder-driven question that we were able to look at there. The next question gets back a little bit to that transmission model. And although we showed that we can transmit the disease, what about other contributing factors that might make elk more susceptible or more likely to get disease? And next, please. And so I'm going to take you back to that same slide I showed you a minute ago and say something just a little bit different about it. And this time, tell you that everything that we did with these animals, we tried to control all the variables other than their exposure to the hooves. And so the animals were in good to fair body condition. They had mineral levels about like what you see in wild elk, and they were treated pretty much, or they were treated the same way. There was no herbicides detected in the soil when we brought that in. And so we didn't have any other contributing factors, and yet the elk still got the disease. So what that tells us is that they can get the disease without other factors, but perhaps what we don't know yet is, could there still be other things that make them more susceptible to disease? And in the future, we'll be able to look at some of those questions. But for right now, one of the next steps we took was looking at mineral status. And minerals are really important to animals for immune function and hoof health. And there's three in particular. Selenium, copper, and zinc are really important for immune function and hoof health. And then there's other minerals that if you have too much of them, they can be toxicants. And those can be found, for example, in some herbicides. And so what we wanted to do was say, what do these elk's minerals, what is these elk's mineral status? And compare ones again with hoof disease and ones without hoof disease. But all I ever get is a hoof. And that's not the best way to look at minerals. Generally, what we do when we look at minerals is we look at the liver, and that's the best tissue to look at. But all we had was feet. So what we did is used hair instead. And hair can be a substitute that we can use for evaluating mineral status. It's not as good as something like a liver, but it could at least tell us if there was anything there that maybe should be investigated further. And what we found was that low selenium may be associated with the occurrence of these abnormal hooves with TAD. At this point, given that we just know it is from hair, we can't say with high certainty, but it's certainly a hypothesis that we're going to investigate further. The other thing that we showed is we didn't see any toxicants that were high in the elk with hoof disease versus those without it. So our next step is to, we're working with wildlife managers in California, and they have liver samples from quite a number of animals, and then they also have TAD positive or negative. And we're going to evaluate the liver minerals the same way that we did with the hair. And I've got a PhD student who's working on that right now. So we should have the answer to that before too long. As far as at this point, I don't have enough confidence in the hair to say for certain that we should go out and be supplementing elk. And that is in part because of a concern if we congregate elk around, say, a salt or mineral block, what we might be doing is increasing the likelihood of transmission when they congregate up. So that's why I want to look in a little bit farther and see what we find with liver. If we continue to find associations with liver, then we might need to think about, well, is there a way that potentially we could provide minerals, but in a way that we wouldn't congregate animals and increase the spread? So stay tuned on that one. Next is, can we treat affected elk? And the tough thing with this disease is, I mentioned there's a similar disease in cattle, bovine digital dermatitis. And even with domestic cattle, and it primarily occurs in dairy cattle, pretty darn uncommon in beef cattle, but very common in dairy cattle. And when the disease occurs in dairy cattle at dairies, they keep the disease in check through things like foot baths and antibiotic treatments. But once it's there, it's really, really challenging to get rid of. You just kind of try to keep it in check. And so even an animal that you have your hands on multiple times a day, we can't get rid of the disease in the domestic animals. And they've also tried to develop a vaccine for cattle and have not been successful. These bacteria are pretty tricky the way they work and make it hard to have a vaccine. So it's super challenging in wildlife because on top of those challenges that you have in domestic animals, again, you have animals that are free ranging over large areas, and also regulations about what we can put into those animals because they're food producing animals. People hunt them and consume them. And because of that, there's regulations about, for example, using antibiotics in them. So we're not going to be able to treat this in a way of kind of traditional veterinary cures, at least in the foreseeable future. Instead, what we're looking at is things like wildlife management actions. One of those could be, again, not quite ready to say it yet, but perhaps once we learn more about minerals, maybe something like mineral supplementation, but I want to hold off on that now. But what we know, I think, enough now to say that some management actions, for example, removing an animal that has the disease to break the chain of transmission. Thinking about how animals congregate and that congregation of animals or movement of animals could make the disease worse. If we congregate animals, that's more likelihood for one animal to give it to another. If we move animals with the disease, we could take it to a whole new area. So we're thinking at the level of wildlife management actions to address this disease. Even though I'm a veterinarian, you know, you're not hearing me talk about more the traditional veterinary type cures. And that's the case with almost all wildlife diseases. It's not unique just to this hoof disease. Next, please. So what have we accomplished? The way that I, as a scientist, am really evaluated is in peer-reviewed scientific publications. And we have a number of those publications. I've summarized their findings as I've answered these questions, and we've got a number more that are that will be published within this year. And those are, I think, important sources to go to understand that is information that has been reviewed by the scientific community and found to be rigorous. And that's different than, say, an observation that I have that might start an inquiry or start a hypothesis. These are controlled or rigorous studies with scientific findings. So we've got publications there, and then also going to scientific meetings where we share this information with colleagues. So for example, wildlife managers in Montana and Colorado and Nevada are looking for this disease as well. And those in other states know, for example, what Washington is doing to address the disease. That's the scientific part, but I think it's just as important that that all of you guys and the people in Washington and other states know what's going on. We do an annual report to the legislature, and that has what we've done both in the calendar year as well as what we're planning to do the coming year. We have quarterly email updates, and if you're kind of tech savvy, you can take a picture of that QR code and it will take you right to our website where you can sign in to receive these quarterly updates if you don't already, or you can just Google WSU Elk Hoof Disease and you can get to our website too, and then sign up for those updates. And I always try to respond to inquiries. I like also hearing ideas because that helps me formulate hypotheses that then we can figure out ways to scientifically test. Next, please. And the last question that I didn't have on the list is, how can you help the research? And particularly if you're a hunter, the most important thing that you can do to help our research is to fill out those surveys that Washington Department of Fish and Wildlife sends, asking things like, did you harvest an elk and where, and did it have hoof abnormalities, and looking at those hooves really well so you're giving a reliable answer. And then sharing the location, which is looked at or recorded in a grid. So we're not stealing, you know, a favorite hunting spot, but we can use that kind of information to look at risk factors about where the disease occurs and where it doesn't. So responding to those surveys is super helpful for our research. Another thing is WDFW has started kind of a pilot incentive program where hunters are encouraged to remove or harvest elk that are limping, have signs of hoof disease. And if we can do that, that's a way to help reduce the transmission. And again, sharing your ideas is always really helpful. And if you hunt, or if you have land, or if you like to hike and find carcasses, if you find elk or see elk that you think we could get the hooves from in those GMUs where we've not yet confirmed the disease, it would be really helpful to get hooves, submit those through WDFW, and then we will look at those in the lab and we can see for certain if the disease does occur in those GMUs. Same with other species. You know, if you see deer and you think they've got the disease, if you can contact WDFW, they can get those hooves to us and we can analyze them in the lab. And that way we'd know if the disease is occurring in other wildlife species. Next, please. So a big thank you. A lot of this work is done by students at WSU. I've got 3 PhD students and 1 master's student who just finished up and a new one coming on, as well as a postdoc and some staff that help out with this. And if there's time and interest, I'm happy to take questions.
I'm going to have to hold myself a while because I got tons of questions, but I think I'll let my colleagues start off with questions, and then we invite the audience. If the audience wants to ask, you would eventually come up to the podium, and we'll talk about it when you come up.
Well, I will tell you, as I was stuck in traffic in Longview, I would have rather been here listening to the comments. There was an accident that involved a bus and injuries, and some work being done on an intersection on Washington Way. So that it caused a little bit of delay for me getting back from the transit board meeting. You showed some of the maps that showed the appearance of some diseased elk in isolated GMUs, and that's just curious. How did they get there? Are the elk migrating across what looked like pretty substantial arid regions to get to the GMUs, or is there just a lack of research in the areas in between?
Yeah, if you wouldn't mind going back to that, either the one with the red pluses and the zeros, or the GMU map. Yeah, that's puzzling, and we don't know how it's moving. With some diseases, we think that moving around carcasses could make the disease leapfrog to new areas. For example, with chronic wasting disease, that's probably how it ends up in new areas. I don't know that it's as big of a risk here. These bacteria don't survive quite as long, but that's always one thing, is moving either carcasses or live animals around can spread disease.
Fortunately, there aren't crosses headed towards Pullman, which would indicate the transportation of carcasses.
Correct. And we're very cautious about that. Yeah, that we keep high biosecurity so we don't accidentally spill it into the area. But, you know, I mean, it could just be a slow leapfrogging. You know, the disease has probably been out there for 20 years. So over time, there can be a slow leapfrogging and movement over time. And in those areas outside of Western Washington and kind of part of the western part of Oregon as well. And well, now a new focus of infection in Northern California as well. A lot of those cases are more sporadic. So I would suspect the disease could have been there for quite some time. And until somebody asked those managers to send in the hooves, they just never looked closely enough to send them in. And that happened both in California and in Idaho, where once we asked them, then we found it. So to answer your question, it could be slowly just spreading a little bit every year for 20 plus years. There's also a possibility that we don't know what the source of this disease is. I'm highly suspect that it's transferred primarily from elk to elk. But when we know we've got a similar disease in cattle, bovine digital dermatitis, could there be spillover events from domestic animals into elk? And maybe that could happen a few areas too. For example, some of the more remote areas where we've seen it, where it's harder to guess where elk would be moving and leapfrogging. Maybe we've had a couple of different spillover events. At this point, we don't know, but some of the work that we're doing with the DNA and looking at the bacteria, I hope that one day we'll know enough to look at like strains or footprints, fingerprints of these bacteria to maybe know if it looks like they're all associated with one initial source that spread out, or if there were multiple places where it started. We're not there yet, but maybe in the future.
And no apparent crossover for horses?
Horses, no, we don't have any evidence that it has crossed over to horses. Canker in horses has a treponeme association, but in the reading that I've done, even for example, in dairies where you have a lot of these bacteria like this around, there's not a strong association with transfer to horses. So not saying it absolutely couldn't happen, but it's not something that has been detected. And horses' feet are quite a bit different, so you'd expect their susceptibility to be a little bit different.
Well, I know we have that herd in Silver Lake area that is wild. So that's one of the reasons I asked. If they're out in the wild, if there's a transmission, but still no evidence of that.
No evidence of that.
That would depend on it, I think, locals being able to identify, oh, yes, there are, if that's really the case. Yeah, great. Thank you. Appreciate that.
Certainly.
Let me see if anybody in the audience would like to come up to the podium and ask questions.
I'm sorry I wasn't looking at you guys when I was speaking. Apparently the microphone's a little bit touchy, so.
It's an awkward situation. I apologize. Yeah.
Yeah, just press that.
There we go.
So if we can go back to the confirmed TAD distribution by GMU, that looks like this.
Just one more slide forward.
Thank you. For the record, my name is Bruce Barnes, and I appreciate the opportunity to speak here. First off, I'd like to know if this was this meeting put in the newspaper? Does anybody know?
It's published. It's sent to the newspaper. I'm not sure how they publish it, but they do have our information on their website, I believe.
Okay, because most people that I know didn't even know about this.
There were not special notices, special ads taken out for this.
There was not a special notice for this, no.
Yeah, so if we're looking for partners, for people that actually enjoy wildlife and hunt it, you'd probably think it'd be a great idea to invite people that do this and support the state Fish and Wildlife with, hunter dollars and buying, firearms and hunting gear and everything else, and are being dramatically affected by these sick elk, because nobody I know wants to eat one of them.
Right.
Yeah, especially me. And if I shoot one, I'm just going to leave it lay there. I'm not going to take it because I'm not going to feed my animal or my family those sick elk. Secondly, we talked about soil samples that we took, and I know that they took the soil sample up there off the Department of Transportation property that was given to Washington Department of Fish and Wildlife. And that area, in my belief, has never been herbicide and pesticide sprayed. So we do have a soil that hadn't been contaminated with these chemicals, right? I'm wondering if we can go into the timberlands that have been sprayed and now take a separate soil sample where they have been herbicide and pesticided and put these elk in a area with that herbicide and pesticided soil, and don't add any lesions to these elk and just see what happens. That would prove a hypothesis that, you know, either the chemicals have an effect or a direct effect. And my honest opinion is these chemicals either have an indirect or a direct effect. Anybody that's got a Weyerhaeuser pass, and I don't know if you've traveled around any of their property at all, the elk don't eat sword ferns, and every elk that I witnessed get shot this year, and the ones that I saw during the winter, are extremely malnutritioned. So when they come into the green up during the spring, that's when you see the major die off of these elk, right? Because they founder themselves on all this greenery that they haven't been able to eat all winter long, right? They've been eating sticks and everything else, right? So I'll elaborate at another time on what I'm doing as a study, because I'm going to prove my hypothesis as well. But we've definitely got a major problem here. And the major problem is the lack of elk, right? Our elk herd is diminished, right? And our Department of Fish and Wildlife is saying, hey, you can go out and hunt these elk that have hoof rot, but yet they've also got a problem that says, or a program that says, we're going to give out master hunter tags to people to go shoot these hoof rotted elk to take them off the surface of the earth, because we think that they're spreading the disease, knowing that these elk are herd animals. They all follow the same trails, right? And as we heard the doctor say, we put chopped up hoof rot stuff in a soil, and these elk contracted this disease, right? So you're never going to stop this disease from spreading, even if you eliminate those, because we don't even know at this point, according to the doctor, that does this live in the soil or does this not live in the soil? Is the soil the contributing factor? Is it not the contributing factor? But we also do know that this is a thing of poor body condition. I mean, these elk on this north side of Mount St. Helens, they look like they're dwarf elk compared to the elk on the south side of Mount St. Helens. The other thing that I have been around this area my whole life, I've grown up here, I've lived here for 58 years, until Olympic Forest Products started spraying on the south side of Mount St. Helens, there was no hoof rot at all, right? And so now that they're spraying over there, elk have started limping in the last 5 years. We also know, because my dad and I were just in Yellowstone Park, that Jackson Hole, Wyoming and Yellowstone Park both have elk that have hoof rot in there, because I talked to their biologists there when I was there. Also,
if I could interrupt you, I
Yes, sir.
think we understand enough that you have some, what I would call certainly strong ideas.
Yeah, questions.
But the reason that we were very enthusiastic 5 years ago is because we have someone who really understands how to isolate things. If I use anecdotes, I can prove almost anything. And we need to have really a scientific basis of isolating things.
Well, absolutely. And that's why I'm asking questions here of,
No, you
Can
weren't
we-
asking questions.
can we
You were
do these
dictating.
things?
Okay, I'll let you answer in a moment. The first part where you say about the advertising, we are doing a pilot program all the time. This is a presentation that is a workshop of the Board of County Commissioners. Typically, you won't find that anyplace else. We're trying to get the public involved and informed. And in that sense, I'm very happy that you're here. In terms of advertising, if you had been to the previous evening presentation, you would have seen a single individual, I think, maybe two in the audience, and now you see 20, let's say. So,
And he was the one who complained that we didn't have evening meetings, so at least he showed up to the one we
Oh,
called
yeah.
for him, yeah.
yeah, so at this point, if there's a scientific answer you'd like to apply,
Yeah, I picked up a couple questions. And the one I want to address first is Yellowstone National Park and Grand Teton National Park. It's really important to note we have not confirmed treponeme-associated hoof disease in those parks. Before I came to Washington State University, I was the chief wildlife veterinarian and was responsible for animal health throughout all of the national parks, including Grand Teton and Yellowstone. What we have in those areas is more traditional foot rot that does occur from time to time. That's a different disease. If you can kind of think about it like a cold and the flu, foot rot is like the cold, and treponeme associated hoof disease is like the flu. They kind of have some similar symptoms, but one's a lot worse than the other.
Well,
And
they sure look the same to me because I
it's a-
talked to an outfitter that had 2 dead elk 2 years ago when I took my parents on a trip there before, and the elk hoof rot looked identical to what it is here.
Well, I'd really encourage them to submit those and get them to a diagnostic lab, because the samples that we've had submitted so far, from Montana in an area not too far from Yellowstone, was traditional foot rot.
Well, I
And
will
those are
search
different
back through my
bacteria.
pictures on my other phone because I use my phones as basically a photo gallery, and I will send you the photos
That'd be
that
fantastic.
I took of those elk.
Yeah, yep, absolutely. I'd love to see those, but as of today, those have not been confirmed. So I'd be really interested to look at those photos, but I also wanted to make it sure that we understand those are not confirmed cases. And that's the only way I will say a case is certain, Tad, is to have it in the lab and to look at it under the microscope.
The other question I have is, when they're doing this stuff in Northern California, right? Are they sending the samples to you, or are they doing their own sampling?
In California, initially, they sent them to us.
Okay.
And then after we diagnosed the disease here, then they started working with their own, the California Animal Health Laboratory Network.
Mhm.
And they are starting to look at their own samples, but they're really good collaborators as well. So we share data and information, but they use the California system.
Well, the reason I'm asking is prior to you being in this program, our Fish and Wildlife mixed and cut up these hooves all on the same table, as you well know, right? And that stuff intermingled. And so it wasn't really a clean room atmosphere to look at this disease, right? Because there was other things associated with it, according to the hoof rot group that was formed prior to you being hired to do this job. The other question I have is that you've been in this job now for 5 years, and this is the first time I've seen any public outreach that you've been at. And that was one of the priorities with taking this over from Fish and Wildlife, is to get outreach to the public, to get the public engaged, right? And ever since this bill was written that Mark Smith and I worked extremely hard on, I haven't seen that outreach at all. And I'm trying to figure out if any of these timber companies are helping out with this, because it's primarily on their land, right? You don't see this on the U.S. Forest Service land. It's only on private timber companies' land. So I'm wondering how much money that they're going to give for research to really put the nail in the coffin on this. You know what I mean? Because nobody likes to see these animals limping around the woods. I certainly
Yeah,
don't.
no, I sure don't like to see them either. And I
Absolutely.
really feel for you guys that look at them every day. It's
Absolutely. It's
hard for me
misery.
to see them.
Yeah,
in
and
the
it's miserable for
pens.
the animal. I couldn't even imagine
But-
walking through the woods with my leg falling off.
Yeah. To answer your question about funding, we are very fortunate that the state of Washington and the legislature has been so generous to support this research. And most of my colleagues spend a good part of their day writing grants to go out and get money to do work, which is they're designing studies, but it's taking away from the time they can do studies. And we are so fortunate that we get this annual funding from the legislature that I don't have to spend time writing the grants. And we are funded by the state of Washington.
And to date, how much money have you received from Rocky Mountain Elk Foundation?
We got $100,000 donation from them for that went toward building our research facility.
All right. Thanks so much. I appreciate you guys putting this on. I think we need to do one of these down in Clark County as well with our Clark County commissioners, because there's a massive population of people that hunt in Clark County. And the other thing about the GMUs, don't rely on Fish and Wildlife's data for people turning these hoofs in and saying they came from this game management unit. And I'll tell you why. No hunter wants anybody knowing what game management unit they hunted in and were successful in, because they print that literature and it floods that hunting unit the next year with hunters. So it's not necessarily true.
You've got a lot of comments. If you document them, you can email them and they'll be looked at. Other questions?
About the bacteria. I'm curious,
Into the microphone, please.
is it present in the soil? Or how did it originate in the soil? Or is it just something that's present in the soil everywhere?
Yeah.
And is it possible that these elk were so overpopulated for a number of years up there that they were just sickly from that alone? And is it possible that just their poor health was one of the causes they could pick up this disease? Like, when you get down, your immune system gets bad, you contract diseases, we do. And that's what I'm curious about.
Yeah, help me remember. Now you had two questions,
Yeah.
and one was, is it in the soil? Is it everywhere?
Yeah.
And right now, we don't have sensitive enough tests to test the soil. So that means the bacteria are there in very low numbers, and we can't detect them using this DNA technique that we use or any other technique. We can't grow them in the lab. But this DNA technique is pretty sensitive, but it's not sensitive enough to detect them in the soil. We've looked at a lot of soil samples that elk with hoof disease have been walking on, and we can't detect it. So it just means that soil is just teeming with bacteria.
Okay.
And so to find these treponemas is challenging. That said, in the future, as we do a little bit more work on the DNA sequencing of the bacteria and finding out which bacteria are the most important, we'll be able to develop primers, which is little DNA sequences that we can then amplify. And it helps us if we can get a soil sample and amplify that one sequence we're interested in, then we can detect the bacteria, even if they're in really low numbers. And so we're working on techniques to do that now. And that's going to be important because that can help us both with diagnostics on the foot when we get that in to make it a much quicker diagnosis, and also to look at soil and say, oh, like around this, wallow or something, do we have a whole bunch? So stand by. I've got a PhD student working on that.
Yeah.
Your second question was more about immune response and like resilience of animals. And I don't have data to say one way or the other, but in other diseases, you're right. There are times when animals are more susceptible and deficiencies they may have and things that make them more susceptible to disease. What our study showed is that they don't have to have those things to get the disease. But what's really hard to figure out is, are they more likely to get the disease if they do have those deficiencies? So one of the things that we're doing is step by step, we have to build these projects. We're working on a reliable transmission model. So the one that I showed you was more of a natural transmission in the soil. So I've got a PhD student who's working on getting a reliable transmission model so we can expose elk in the pens to material and they'll have disease within, say, like 4 to 6 weeks. Once we have that reliable transmission model, then the next step would be you could have one group of elk that you feed up and they're nice and fat and you give them Zinpro mineral supplements, and just do everything that you can do to think of to really increase their immunity and their resiliency. And then you've got another group that's really skinny and mineral deficient and I would say exposed to herbicides, except there's so many different combinations and concentrations matter that was going to be a hard one to test. But we could at least have different groups and see if the susceptibility is different. Maybe one group only gets to those really early little small lesions and the other one gets to the really bad lesions. Maybe they respond the same. We don't know, but it's a good hypothesis, and over time we'll try to test that as best we can.
Mm.
And then also doing other laboratory work to learn more about the immune response, to where eventually we could look at animals in the wild and look at their immune response. That's only one component of the disease, though. That's like immunity to one thing. And then like you brought up, the resilience of the animal, their body condition, and just kind of how they can fight things off. That's much harder to get at.
Well, the herbicide thing is, personally, I don't think that spraying their hoof is going to give them hoof rot. It's the result of the herbicide. These clear cuts don't have any nutritional value at all. They're dead for years. No bacteria in the soils, no mushrooms growing back. You know, I'm not a biologist, obviously, but just see the blackberries are dead, the huckleberries are dead. The only thing that grows back are noxious weeds and those fir trees grow crazy, you know, but that's what the whole idea is too. But so that just leads me to believe that there's a possibility that they're not getting enough nutrition to maintain their health, especially we were really overpopulated for years. Everybody loved those elk to death. And we couldn't even hunt them up there for years until they started dying off. Then they started opening up the hunting again. So now the population's just gone extremely down. I used to have 40 head on our property all the time. Now I haven't seen an elk for a couple of years. So, you know, the population's going away. So that's part of the purpose, I guess, to get rid of it too, reduce the population. It's happening. So anyway, thank you very much.
Yeah, one thing that I thought of also to address your question is, certainly here elk are in much poorer condition. In Northern California, though, the biologists that I talked to there say that those elk with hoof disease tend to be in like pretty good to very good body condition. So again, it's not to say that elk that are skinny might not be more susceptible. I'm just making the point that even fat elk can get the disease.
Yeah.
But it's hard to figure out all those different factors.
Well, it started here, I think.
It seems to have, yes.
Thanks, I appreciate it.
Thank you.
Good evening, gentlemen. Thank you very much for holding this meeting. Mark Smith, Dr. Wild, thank you for coming this evening. I think you did a real good job covering, you know, where we're at to this point. Just a couple questions I have for you is, number one, we still have you accomplished testing a live animal in this area to your satisfaction. I know there's been some problems when in the past working together, we've tried to get a sample of an animal here. For a number of different reasons, we don't have to go in details, but have we accomplished that to your satisfaction of getting the samples of the liver, the animal, the blood, and that from an actual elk in the area that's affected the most right here?
Yes, we've been able to do what we need to do, and we always need more hooves that get into ultra cold -80 freezer within 24 hours of death for our transmission studies. That's a challenging thing to do, and it's an ongoing need because we don't want them to sit in the freezer for a long time. But to this point, yeah, we've gotten the samples that we need. And it's great to work with other collaborators as well. For example, California has been a great collaborator too.
Okay. And one other comment that, well, just two quick ones. The overpopulation that we saw of the elk, you know, when you overpopulate in a herd, sometimes the genetics get a little bit lower.
Mhm.
And I haven't heard you comment about the genetic situation because you know, the
Yeah.
elk all looked like the same for many years, you know, and especially the bulls and that. So have you seen a change in that?
That's a great question, Mark. And we're not there yet, but every hoof that comes in that we do diagnostics on, we take a chunk of tissue and that will give us host DNA. And it's farther down the path but eventually we'll be able to start looking at things. It's called like a genome-wide association study and see what's different about this elk than that elk. And that's something, for example, we've done with chronic wasting disease and found different genotypes, some which are more susceptible, some are less susceptible. There may be something very similar to that going on in elk. It's technical work. It's what's the word I'm looking for? Complicated work, but we've got the samples. So we're planning ahead to do that, but have not done that yet.
Great. And the last one is, what does the effect seem to be, if any, on the birthing rates or the young calves are born with it or not? Do we know any of that at this time, how it affects their calving?
As far as if calves are born with it, I can't say for certain one way or the other, but from what we've seen at postmortem examination of elk with the disease, it's not the kind of disease that's probably transferred in utero, transferred directly to the fetus, because we don't have evidence of a lot of those bacteria circulating around in the blood and going other places. I can't tell you that for certain, but that just as a veterinarian makes sense to me. However, of course, as soon as that calf drops, if its mom's got the disease and it's standing there right next to mom, not only stepping in the same footsteps, but probably, stepping on her feet, very early in the disease, you could very likely get transmission. I don't know, Kyle Garrison, WDFW is doing a survival study. I don't know if you had any additional comments on calving.
You should come up, but otherwise, this is being recorded. You'll be able to see this on the.
Yeah, and
College
since
County
Kyle's here, I
website.
didn't see him sitting behind me. I didn't look either. The question is too, I've seen a lot less calves because we're seeing less elk, but do you think that the cow elk are affected that have hoof disease, not allowing a bull to mount them to be able to be bred? I mean, basically, because we see a lot fewer calves, even though we're seeing the same herds in areas. So if you can address that, I appreciate it.
Yeah. So I think, Mark, your first question to answer is, do we see an association with like reduced productivity or reduced pregnancy rate with the disease? And I'll say the easy answer is to say yes, based on our survival study that we did in Mount St. Helens. That was from 2015 to 2019. We're currently wrapping up that reporting process. Animals with the disease have much lower pregnancy rates. So we capture them, we look at them, we're able to do ultrasound blood testing to get that pregnancy status. So that's around 50% across the board for animals that have the disease, as opposed to more, you would really expect that in an adult female elk to be more like 90%. So it's pretty tangible effect there. That's a depressed pregnancy rate. The problem with linking it directly to the disease causing that is that we've talked a little bit about nutrition. So it's a bit of a chicken or the egg, where clearly the animals that have the disease are in worse nutritional condition. We have those body measurements. And we know from other research that's not associated with this disease, that when you have poor nutritional condition, you're going to have depressed pregnancy rates. And so there's part of that going on, but the question really is that animal in poor nutritional condition because of the disease, or is it that it was in poor nutritional condition and it got the disease? And it's probably a little bit of both. Those things probably interact. So both of those things might be able to be true. So we do know that we have depressed pregnancy rates. And then the next part of that is, if you assume that that effect might be ubiquitous across any elk population that's affected by the disease, although Mount St. Helens is a little bit of a special case because of that nutritional component, if you have to pay attention to sort of what the prevalence rates are. So Mount St. Helens has, at least in Washington, it has the highest level of prevalence. So more animals are affected by the disease than any other elk population. And so it's over the last 5 years, based on our hunter reporting, which isn't exactly a direct estimate of true prevalence, and it's an index. It's sort of a informative. It tracks probably with natural prevalence, but there's some limitations there. It's about 30% of the animals that are reported harvested are also reported by the hunter to have an abnormality on their hooves. And so that's the index we have for does that animal have that disease or not. It's pretty reliable. There's some caveats there. In other populations like the Willapa Hills overall, it's more like 10% over that period of time. And so it makes sense that given that we have a higher number of animals that are affected by the disease, lower pregnancy rate in that group, in that slice of the pie, that we're having less calves even just being produced, let alone surviving, given those other outstanding nutritional limitations that are occurring in the population. And then the population of adult females that don't have the disease are also exhibiting depressed pregnancy rates. Those are normally around 75% from what we found from our most recent research, which again is lower than what we would expect and anticipate in pretty much any other free-ranging elk population. Does that about cover it, Mark?
Oh, wait, before you go. Your answer seemed to assume that the rate of intercourse was assumed to be the same regardless of the mechanical condition of the animal. Is that true?
So getting kind of what Mark was asking, like, are they even able to be receptive? I don't know if I've heard any suggestion that like a cow might sort of, reject a male because she has the disease, some sort of function of being in pain or, can't, isn't stable enough or something like that. I would be a little surprised because of course, that is the name of the game, right? Maximizing their fitness, maximizing their reproductive output. They'll pretty much trade off anything to be able to do that. But, there is the possibility that the animal, again, in poor nutritional condition, maybe isn't able to actually go into estrus. And so then she wouldn't be receptive at all. But the mechanics of it, I'm not sure that I've heard any other anecdote that maybe they're not able to pull it off.
It's confounded, of course, because we've all heard the not now I have a headache, and we don't know whether that's a hoof ache or not.
Yeah.
And I wouldn't put the blame only on the cow. I'm not sure about the bull.
Sure. I would also still kind of rest on it, those bulls, they have one thing on their mind, and I don't know if a hoof is going to stop them. I mean, any of these folks out here, anybody who's hunted elk and seen these animals, they're very, very tough. And, when you fly around, you have these animals that have the disease, their ability to move on the landscape, even without a hoof, that's just essentially a bloody stump, and they're still at a full sprint. These are extremely tough animals. So.
Right. The point, though, is then you are assuming that the rate of intercourse is the same in your answer, and that would need to be proven.
Yeah, that would require further study to have a definitive answer there. Go ahead.
Make sure it gets recorded. Also having a few years of experience in witnessing the disease and seeing it and being in the location that I'm at, there's one factor that I think is apparent in almost of the GMUs that you see in that. And I would like to know if you've considered, but it's the stress. Okay, we start hunting these animals in September over the same area, the same GMUs with, hunters, archery, muzzleloader, moderate firearm, then archery again, muzzleloader, special hunt, special permits, to where these animals are not just in a normal season of hunting, they're stressed for probably 4 to 5 months of the year. Is stress a factor that you've considered? Because your animals are in a nice cage, they're getting fed, they're in a field, they're not under any pursuit of stress. And anything that has stress usually is stressed.
Yeah. We have not looked at cortisol levels or other indicators of stress in the animals. As much as I would like to say our animals are not stressed in captivity, we're taking wild animals when they're about 9 months old and putting them behind a fence. So there's probably some, but I hear what you're saying, it's a lot about like disturbance too, just keeping animals moving around, not having time to feed. And to answer your question directly, no, it's a hard one. It's a hard one to get at, but again, we reproduce the disease without additional stress other than the fact that these are wild animals now kept in a little pen. Yeah, I would think a lot, as much of anything. I did my PhD on cortisol and stress in bighorn sheep. And as much as anything, I might think disturbance and like messing with time that it takes to be a successful animal, enough time to eat and sleep and do everything, as much as stress but stress can have an influence on the immune system
Yeah,
as
I think
well.
this, my theory would be the stress would be the disturbance, so.
The disturbance part, yeah, yeah. Mhm.
I'll answer that real quickly. In general, we are concerned about disturbance. We know there's a large body of literature on elk about the effects of disturbance. That's why we have things like winter closures on wildlife areas and access restrictions and things outside of those hunting seasons. So it is on our radar. We are concerned about it, but that also is balanced against our mandate to providing a maximum recreational opportunity, and our hunting public consistently stresses that opportunity to go out, recreate, to pursue elk across a variety of hunting seasons is really important to them. And so that's us balancing these different values and different asks of a resource. So, and I'll step away now.
My question is dealing with possible solutions to an elk that has a problem. Have you done any testing to see elk's got the hoof disease? What works to cure that disease? Have you done any testing? You talked about foot baths, possibility of supplementation, antibiotics. Have you done any testing to see, hey, if we got this elk, we got him captive, can we inoculate him? Can we give him foot baths? Have you done any testing of that?
Yeah, that's a good question. The short answer is no. I did have it written into our last study plan, that transmission study that I showed you, where 4 out of the 4 elk got the moderately severe lesions. It was in our study plan where we could have done an antibiotic treatment and seeing if we could reverse it. So I was left with, do I do that, which in a field situation has very little application. So even if I showed that we could treat them with antibiotics, it's not something we could do in the wild. And it would also mess up the samples that we would need to see if the transmission study worked. Because I would suspect it would decrease the lesions. I would suspect it would work. And by doing that, it would make us not be able to completely, purely interpret the results without the antibiotics. Does that make sense? So I wanted to really protect that first study and the findings so we were certain about them. In the future, we could do that, but it doesn't make a lot of application type sense to me because it's not something we could do in the wild. The reason I thought about doing it was to say, do we know that bacteria are really the cause of all this? Because if we treat with antibiotics and the lesions go away, then that's another layer of evidence that it's about the bacteria.
Okay, let me give you a scenario. Free ranging elk, 8-9 months of the year, going to be hard to do anything as far as treating them. But during the winter months, I look at a herd in our local area that comes to my neighbor's field, 250 acres, grass. They stay there for 4 months. They're eating grass. They're not eating the woody stalks out in the brush. And they love it and they're not harassed. And so they're concentrated so that if there was any treatment that could be done, they got to go to water. There's a possibility of putting a foot bath in there. I tried 5 years ago putting a mineral block out in with permission from my neighbor that had extra selenium and copper in it based on a discussion I had with Kristen Mansfield. And she said, at that time, we think that might help, but we don't know unless the hoof is so very bad that probably won't be cured. In 3 months, they had a 50-pound block down to 25 pounds. And did the elk get help or not? There's no way of knowing whether or not that helped them or not. I'm looking at the Mount St. Helens high area. A lot of the elk concentrate during the winter time in the North Toutle Valley. Another field farther up the road from me, maybe 30 elk that winter there pretty much steady. The elk are concentrated enough that if you had a mineral block with antibacteria in it, mineral supplementation, maybe a place for a foot bath. These are possibilities because the elk are concentrated during that time of the year. During the rest of the year, they're also scattered that it would be impractical. But during the winter months, I'm suggesting that might be a way of if you know what the treatment is that helps either prevent or cure the disease, this might be a way of at least getting something started.
Yeah.
So I suggest if you have some means of testing to see what's going to work, that might be, the winter months might be the opportunity to put some of this stuff in place.
Yeah, I mean, I appreciate your problem solving approach and thinking about it. I think logistically, it's going to be really challenging. Number one, getting wild animals to walk through a foot bath, but these chemicals that are used are not environmentally friendly. And in cattle, they have them walk through there repeatedly. And remember, I mentioned that they can keep the disease kind of under control, but not cured. And with antibiotics, there's also laws about what I as a veterinarian can put into a food producing animal and when I can do it and how I have to make sure I keep it out of the human food chain. So there's challenges there. But a really important thing you brought up was this congregation, and that's just a hotbed for transmission. It's like it's a daycare, with respiratory disease going around. If we are looking at minerals, if you're going to put out minerals, I'd ask you to do one thing, and that is try to disperse them and put them in the driest place possible. So animals aren't standing in mud to lick it. I know people want to do something, and I'm saying I'm not quite ready to say, yeah, go out and put out mineral blocks. But if it's something you want to do, just try to do it in as dry of area as possible and move it around so you're not really causing like a hotbed of infection.
Yeah. Okay. Well, anyway, that's my comment.
Appreciate it.
Good evening, and thanks for the opportunity to attend. And I think the outreach is something that is working and evidence of this meeting, and I think more is needed. I'd like to compliment the research effort. I think it's fundamental to understand the transmission to do with how it spreads. And if there's any future work done on resistance, that is elk get it later in their life, or actual immunity, I think would be important down the road after some fundamental stuff. I'd like to ask a question of the WDFW representative locally in Cowlitz County and in Lewis County, in those game management units, in the surveys and the estimates of population, what are the elk doing in terms of, are they stable? Are they still declining? And if you could address your comments in volcanic soils around Mount St. Helens and in sedimentary soils in the Willapa Hills. Thank you.
All right, I'll do the best that I can. And I didn't introduce myself earlier. My name is Kyle Garrison. I'm the ungulate section manager for Washington Department of Fish and Wildlife. I am based in Olympia, so my local knowledge is somewhat limited as opposed to what our local district biologist, Eric Holman, would have been able to offer. But I did bring our statistics here of our surveys. So we fly our surveys usually on an every other year basis. Those are aerial surveys. One thing to note is that when we do those surveys, Western Washington is a unique landscape for elk where when you're flying in a helicopter, you can imagine you're looking at a lot of trees and not a lot of elk. And so we have what is called the detectability issue. And so where we can see elk are generally more open areas, and that's a subset of the landscape. And so what our estimates are more of an index of abundance rather than a true abundance estimate across the entire landscape. And so where that's most apparent in particular is our estimates for the Mount St. Helens population. We're really only able to fly 5 core units, what we call the 5 core units of the Mount St. Helens population that is on the Toutle Valley, that is on the mud flow of Mount St. Helens, where historically we've had the highest population densities there on the Toutle River. So for those of you that are from this area, spend a lot of time hunting these elk, you probably know that about 12 to 15 years ago, there were a lot more elk in this area. And the department took some purposeful steps to try to bring that population down. We had some indications there were nutritional limitations, it's been mentioned earlier. And then the disease came in as we were lowering that population. 2011, 2012, we knew the disease had entered the Mount St. Helens core units in 2013. So we ceased that intentional population reduction, but unfortunately the population did continue to go down. So here's just some statistics. Our peak in 2011 was roughly around 5,000 elk that we had estimated in those 5 core units. Those are GMUs 520, 522, 524, 550, and 556. So we've seen that population continue to dwindle down. I wish I could show you this here on the screen here, but just to get to the point here in 2022, we estimate there's roughly about 1,500 elk. So it's a pretty substantial reduction. We have about a third of the elk that we did 12 years ago or so. So that is the point the preamble there is to emphasize that there was a purposeful reduction. We achieved that, reducing the population by about 1/3, trying to get it back in line. We had overwinter die-offs that you wouldn't normally have otherwise seen in this climate. We knew there was an issue around 2008, 2009. And then the disease came on the landscape. And then we had some pretty intense winters. And all that nutritional limitation, the effects of the disease, these environmental conditions have all kind of come together to coalesce, have this consistent decline. The good news, if there is a silver lining, 2019 was the lowest we've ever seen it. It was about 1,300 elk. So we have a slight tick up from this most recent estimate. So I would indicate that I would suggest that that's probably indicative of stability at the current low level, at probably a historically low level, at least in terms of when we've been able to monitor those core units of Mount St. Helens. Now in the Willapa Hills, we separate this population into a north and a south. From a management perspective, it's a little bit easier. The population, is ubiquitous. It doesn't recognize kind of our split there, but it's also a way for us to survey this population a little bit more logistically easy because it is such a large landscape. It's difficult to do in a single survey. So Willapa Hills herd demonstrates a lot more consistent population levels. Some of the same caveats exist about that detection problem, and we're only seeing elk where you can really count them. But nonetheless, from 2016, when we initiated our siteability surveys, we had about a little over 3,500 elk in the South Willapa Hills, which would include this area down here. In 2023, our most recent survey, we had just under 3,000 elk. So we have seen a modest decline there, but it's not at the magnitude that we saw in the Mount St. Helens elk population. In the North Willapa, we've actually seen a bit of the inverse. So we began that survey in 2017, had a little over 1,000 elk that were estimated on those surveys. 2023, we've got about 1,500 elk that we estimated there. So again, just some modest growth there. But overall, if you combine those, I would consider that a relatively stable elk population, if a modest decline. And I think that covered all the questions. Are you hunting at the same levels? Or have you reduced the number of hunting days and opportunities, knowing that we should Yeah, so the question
you-
was, do we manage these populations differently in terms of their harvest regulations? Is there more take from one population or the other? So in Washington, we manage our elk and most ungulate hunting, deer and elk hunting. We're an over-the-counter general season state. So you go buy a license if you're an eligible hunter, you can get an elk tag and you can go hunt, as opposed to another state system that might be restricted by having a permit quota. What we tend to do in Washington is focus our general season harvest on the male subcomponent of the population. That's a component that can take a lot of harvest pressure, and the population can still perform because there's only really one male needed for a number of females. That's just the reproductive ecology. So our general seasons are pointed mostly at that male component. And we don't have a difference really between those two. We do have some GMUs in the Mount St. Helens population that have more restrictive general seasons or no general season, and they are by permit only. They're managed more for a quality experience based on the input that we had from that local, the hunting community that really advocated for having a variety of hunting opportunities there. That wasn't just a general season opportunity. But what's more important really for a population's performance over time and in terms of managing the numbers of animals that are out there is the antlerless take, the cow take. We tend to restrict most cow take through special permits, at least for modern firearm tags. There are some cow seasons in, say, an early archery or a late archery season that perform, you go out and you can hunt. So we don't have relatively little control over the take there, but it tends to be pretty conservative and pretty stable. So to answer your question, Mark, we have pulled back on our antlerless harvest in the Mount St. Helens elk population in particular. There's almost no antlerless take that is occurring that is not associated with conflict mitigation. So that's agricultural conflict mitigation. A lot of our antlerless harvest is either issued to reduce a population, which is of course not the goal we have in Mount St. Helens, or more commonly, it's to address agricultural damage issues. In the Willapa Hills, because that population does perform pretty well, it has pretty good stability in our surveys and in our harvest statistics. We do have more antlerless permit opportunity, but as a rule, that antlerless permit opportunity is always conservative.
Chip. Is it truly a question?
This follow-up question, yeah. If we're concerned about elk congregating, why are we still having a feeding program at the Oak Creek feeding station? Because that is a massive congregation of elk. Firstly, secondly, anybody that has a groundwater well on their property has to have it tested by the Department of Ecology. And it tells any disease or anything that is in that water so it's drinkable, right? Why couldn't we take some of these elk areas that have a huge congregation of elk that have hoof rot, and we could make a slurry, just like taking that bottle of water right there, and we just run that dirt through that water after we test the water to see what is in that water. And then we compare it to after we run the dirt through that water. And that would be your way to prove if the disease is in the soil. Just a suggestion. Thank you very much.
It's still, there's still a lot of bacteria there. So there's millions of bacteria in the soil. And so that's why we've got to use different techniques to amplify to find the ones we're interested in. The first part of your question was, why are we feeding? That is a management decision that, managers have to make a decision based on a lot of different factors, and Kyle can speak to that. As a veterinarian, that's something that I do not promote. I don't promote it on the National Elk Refuge, where you have chronic wasting coming in. I don't promote it anywhere because it's a way to promote disease. However, my vote as a veterinarian is not the only vote that matters. Managers have to take in a lot of other votes. And so Kyle, I don't know if you want to speak to that.
Yeah, it's a really good question, Bruce. And it's it's always a challenging one. Of course, wildlife feeding, we advocate,
Could you do me a favor?
Yeah.
Could you move that laptop so that you're in the
Oh,
picture?
yeah. Absolutely. Trying to give,
Okay, thank you.
kind of shoulders here. So we as a department tend to dissuade folks from feeding animals. We recognize Margaret's really covered very well some of the issues and the problems with feeding wild animals. And so it is a bit awkward for us to have a wildlife feeding operation there in central Washington. We have multiple feed sites. And what Margaret just mentioned, I think, is a pretty good articulation of why we have that. So we are balancing multiple values, multiple socio-political pressures and preferences from a wide variety of user groups and Washingtonians. And so we have on one hand a desire to have a large, robust population of elk in central Washington. But on the other hand, as humans, we've developed their historic winter range and turned that into agricultural production. And so when we have a large population of elk, they've got to come down, they're in their winter range, they're going into those orchards, they're going into that agricultural area, they're causing a tremendous amount of damage. And so in order to mitigate that, at least in part, we have to feed those elk to keep them out of those areas to mitigate that damage while also maintaining that population. Now, as a department, we're also moving toward trying to find alternatives to that, and we're trying to find unique alternatives to move away from that feeding. And I'll note that it isn't an every year thing, and some years we don't feed because the winters aren't that bad. Some years we only feed a little bit because it's not bad. Some winters we feed a lot because they're really intense. I can't comment on last year's winter, but the preceding winter, I don't think we really fed at all. Or if we fed, it wasn't until the end of February. So it's a difficult problem and one that we're trying to work toward moving away from, but not a problem that has a simple solution that you can just rip the Band-Aid off and move forward with.
Thank you. So maybe Dr. Wild, I can ask you a couple of questions, although my list is large. I'll interview at time. How's your time? Are
Doing
you-
okay, sure.
Okay. I'm going to be more on a focused scientific kind of approach. You mentioned earlier that there is treponema and 3 other bacteria. When you ground the hooves, did you try and isolate the bacteria and say, this fellow is going to try the treponema only and this one?
Yeah, that's Koch's postulate, right? Where you take a certain bacteria or pathogen and expose an animal and then try to get it out. We did not do that. And that was for a couple of reasons. One is they've tried to do that in cattle with bovine digital dermatitis to just, number one, it's really hard to isolate just the treponema. It takes special culture techniques. And when you do it, you don't get all the different treponema, you just get a couple. And they've tried that in cattle where they culture a couple of types of these treponema and then try to give bovine digital dermatitis to cattle. And they've primarily failed. And so that has led to the hypothesis that bovine digital dermatitis is a polybacterial disease, that either you have to have a bunch of different types of treponema, or you have to have treponema along with these other bacteria like Mycoplasma, Fusobacterium, Porphyromonas, and they work as a team. And one of them does one job and another one does other jobs. And I would suspect that we would find the same thing with Elk Hoof disease. It's probably a team effort. And from a manager standpoint, it really doesn't matter as much that we know precisely which one. I think we could go down a rabbit hole of years of research to try to figure that out without it moving management, ability to manage the disease forward. So for those reasons, we decided to leave it as a group of bacteria. The next study that's being done by my PhD student now, though, is she's going to try to enrich the treponema in that inoculum to see if we can get a more effective inoculum, and that might give us some indication of how important that is. So we're kind of skirting around the edge of your question, but I don't want to go to where we go one organism at a
Oh, I like
time.
the answer, the combinatorics. I mean, even if you found something out, there are other more important things to find out at the moment before you spend your effort there. I understand.
Right.
If I gave you a blood sample from an elk that was diseased and one that was not diseased, but you had no idea, I just gave them to you, would you be able to say this elk is diseased and this one's not? It's my backhanded way of asking, is this in the blood system?
We have not detected any abnormalities in the blood system. So, I haven't actually taken DNA out of blood, tried to take DNA out of blood and submitted it, but we've looked at tissues
Mhm.
where the organism should be, and we don't get them. So, and when we look at the pathology of the disease, it's not as though those bacteria are frequently going all over the body and setting up infections elsewhere.
Yeah,
So the answer's no. Now, eventually, maybe I could be able to tell you if it had been exposed to the disease, if we can get a more reliable way to look at antibodies in the blood. But right now, we don't have a reliable way to do that. And again, it doesn't tell you if they have the disease, only if they've been exposed to it.
yeah. Does that mean that if you're trying to find a cure, you're really looking at something that is of a topical nature then?
Well, again, in cattle, they use something that's more topical. So whether it be the foot bath or the antibiotics. In wildlife, I just don't feel that the technology we have now, that's logistically feasible, which is why I go back. to
Yeah,
more
I'm
managed.
not asking about the feasibility of
Yeah.
doing it, but if you had everything under control, you'd look for a topical solution rather than a.
Yes, the disease really is isolated primarily to the feet. And I do understand that, as animals get debilitated, they get other kinds of infection. But just the foot disease initially
Right.
is isolated to the feet.
So then if I got you two hooves from two diseased animals, and I told you nothing else, could you tell me what in that map with the circles and crosses, could you tell me where those two hooves came from?
At this point, no. There's ways to get at that through the host genetics.
Right.
And that would take another line of inquiry. Another way to do it, Kelly, if you could just start flipping through the slides, there's one of them that's the state of Washington and it's got like 5 colors on it. And maybe it probably also has Idaho and Oregon. And it's got like 5 different colors on it. Anyway, what that map shows is I've got one of my PhD students and the work that she's doing with looking at the bacteria through the DNA that's there. We've got, I can't remember how many locations. It's probably like 6 different groups geographically. And she's going to compare those and see, are we seeing the same types of bacteria in northern Idaho as we're seeing in southwest Washington, as we're it's at the very end. It's beyond the end of my presentation in the extra slides. And she's going to try to compare and see, do we have similar bacteria in these 6 geographic areas? Our hypothesis is they probably are the same, but we will have to test to find out if that's the case.
As you looked at the
There it
bacteria,
is. So those are the different areas that she's going to
Yeah.
compare.
as you looked at bacteria, do you have a sense for the viability of the bacteria outside of the hoof? Because there've been a lot of questions of it. Sounds
Right.
like I can go out there and 10 years from now, I can find the bacteria was hibernating. That kind of question.
Yeah. When we look at the DNA, we can't tell if the bacteria is dead or alive. We can tell the proportion in the sample, but we can't tell if it's dead or alive. In the reading that I've done in my experience, these treponema, if that is what we're primarily interested in, don't survive super well in the soil. I say the soil is likely a short-term reservoir. So maybe a matter of hours, maybe a matter of a couple of days, but not weeks, I don't think, not months, and certainly not years. And that's very different from, for example, chronic wasting disease, which really does that prion, infectious prion stays in the soil and stays infective for years, decade. I would hypothesize that these are only in the soil and viable and infectious for a short period of time. But we have not done the studies with these particular organisms to know for certain yet.
Okay, so back in the old days, if a herd of cattle was found to have, what is it, anthrax maybe, or sheep, and then they would just take the whole herd and bury them, throw them in a lime pit. Okay, a question on, of the ones that you inoculated, you said you had to put down 4 of them and the 5th one survived. Did you learn anything from that?
We took a lot of samples from him. I can't tell you today why he recovered, why he got bad and then he recovered, but we have a lot of samples from him. And in the future, I hope that he will be one of the animals that we'll be able to look at to give us clues as to what might give us more resilience or immunity or genetic diversity and ability to fight disease. But right now, we just have lots of little pieces of him that we hope to learn from.
You mentioned vaccination before, it not being viable in some sense. If you try to reinfect him, it'd be an interesting, I apologize to that poor animal, sorry.
No, you bring up an interesting point. And in cattle, again, if we're using cattle as a model for hoof disease, they can get the disease over and over and over. They have the disease, you treat them, you stop treating them, they get it again, they get it again, they'll get it again. So my suspicion is that probably the same thing could happen with elk.
Okay. And the question was asked by one of the gentlemen about the origin of the disease, and that's like, where did any disease come from in the first place? So I wouldn't want to pursue that too much.
I'll say a couple things about that.
Okay.
I've got a postdoc right now who is working on putting together all the DNA in a sample to reconstruct genomes of bacteria. And it's too early to present that work. We haven't submitted it for publication, but he is detecting what appears to be genomes of new bacteria. We're discovering new bacteria that are like these treponema, but not exactly like the ones that have been described or sequenced before. Over time, what I'm hopeful of is that we'll be able, and we can look at phylogenetic trees, just like you can look at the evolution of any species and say that elk and deer are more closely related than elk and deer are related to elephants. But, and so we're doing some of that with the bacteria. I don't know how far it's going to get us, but I hope it will get us kind of to an idea if these are more elk adapted or if they're the same thing and maybe came from cattle, or give us a little bit of idea of where they came from. But that's always a super challenging question. You're right.
Yeah.
And really, where does it get you? I mean, maybe it helps you from making
You are where
the
you
same
are.
mistake again.
Yeah.
You're still where you are today.
And this is more of probably a silly mechanical question. Doing a liver biopsy, can you not take one from a live animal without destroying the animal?
It can be done. It is risky. And again, in the wild, it is something that is a pretty invasive thing to do. So it's a lot easier just to do it on hunted animals. Hunters are a wealth of samples. If you can organize them and get them to handle samples correctly and get them to the right place. And so fortunately, we've got a nice set of samples that we can use from California. And it's best, I think, to get those harvested animals.
Okay,
Lot
thank you.
easier on the animals than putting them through a liver biopsy.
Looks like we have a gentleman that wants to ask a question.
All right.
Yeah, please. I figured by now you had been tired of my questions.
Thank you so much for coming and presenting. My question, I guess, is about the path forward from where we are now. I appreciate that the scientific process takes a fair bit of time to, build a careful understanding, and it sounds like we have learned some things from this process. But some of your comments made it sound like the science isn't yet at the forefront of the management decisions, like the discussion about the animal feeding, and it sounds like your recommendations around avoiding congregation of animals. So what I'm driving at is it sounds like from the people in the room that there's a real concern about the population of elk in our community and the opportunity to use what we're learning to make things better and make that population healthier. So in your estimation of all of the research that you have planned out, could you maybe give any indication of what are the next steps? When do you see the science maturing enough that it has more of an impact on the management decisions? And what do we do in our community along the way while we're waiting for the science to answer some of these questions?
Yeah, that's really the big question, isn't it? That's a tough one. And people in different communities have different questions as well. So last night I was up in Skagit County, and those folks had very different kinds of concerns. They were concerned about too many elk and a lot of concern about elk livestock interactions. So I'm kind of trying to balance how I set priorities with stakeholders like that have one set of questions. And then you guys who are seeing much more impact of the disease and are concerned about not having enough elk. So where does that take our research? If you want to know where we're going, there is a report to the legislature that I write every year. And the first part of that's what we did the previous year. So what we did in 2022. And the next one is what we're doing in 2023 and moving forward. So it gives you a little bit more of a step by step. And they all fall into 4 categories. And you're right, it is more about basic foundational information, but we're trying to learn about the organisms in the laboratory. So this, like the DNA work with the bacteria, in the pens where we're looking at transmission and maybe get at some susceptibility. We're also we're doing computer modeling of risk factors. I've got a PhD student working on that. And then the last one is on the human dimensions, the social science and kind of stakeholder driven research. So it's a very broad approach that we're taking. And what I'm trying to do is take on the questions that I think are most tractable. Some people say the low hanging fruit, that are the easiest to address and have the most management impact. And just take those one at a time. And I think one of the big things that we've learned is that because we do have strong evidence this is an infectious transmissible disease, that, for example, when I have my conversations with Washington Department of Fish and Wildlife, what could we do maybe to increase removal of those animals? They've got the incentive program going. Could we go farther with that? Are there other ways that we could try to reduce the number of affected animals? To do that, it's not only a biological question, but we'd have to think about that socially, because folks like you guys that see those animals every day might say, yeah, I mean, just from an animal welfare standpoint, let's take them out of their misery. Other people who haven't seen that would be saying, oh man, don't kill them if there's only a few of them that are lame. But from a management standpoint, we know that's the best time to remove animals. It's early in the disease course, because by the time you have a whole bunch of affected animals, you can't really remove them without having a huge impact.
Yeah, I mean, it seems like you're almost driving towards some like policy or recommendations that could be used by communities. I don't know if it's a partnership with Fish and Wildlife
Yeah.
to have more detection so that it's easier to identify when the disease is appearing in a place and apply some of those methods. Who are the local partners or sorts of organizations that can lead the charge in partnership with taking and trying to apply some of this research? Do you have any thoughts on, is it the county's responsibility? Is it other types of jurisdictions that can help develop more of these things that we can actually be proactive and not necessarily have to wait for all of the science to come in?
Boy, you asked the hard questions. Yeah. What I try to do, I guess, is through networking. I don't have one approach, but just trying to meet people who know other people and help us network and get to different groups, because we know from some of the survey work that we've done that different groups have very different opinions about things. And so it's got to be a long conversation and listening to different perspectives and sharing information. And that's something I don't feel like we're at that point yet, that we're not doing. But as we get more and more data that we have confidence in, then I think we can have those conversations, because when we have those conversations and everyone's coming from this is what I see, this is what I see, then it's hard to move forward. But when we can build trust and have rigorous science, that's what I think how we can build on those conversations. And maybe it's time that we can start having more of those. And if you have ideas, I'm very open and would love to hear them.
Thanks. And just any guess on the timeline to get to that kind of point where you feel there's enough evidence to really inform those conversations?
I think we're probably there for some locations. And I have to say that the Southwest Washington is one of the more challenging ones, because there is a high prevalence. There is a low number of elk. But maybe someplace like in the North Cascades, where there's apparently too many elk and people are more concerned about transmission, maybe there would be more, at least the limited number of people that I've talked to, maybe there would be more support for lethal removal. Maybe we need to identify small locations, kind of like what WDFW is doing with the incentive program as a pilot, and find pilot areas where we can try some different things, rather than thinking we can do it on a broad scale. Try to do it and learn on a small scale. And I think we're about there, where we could probably start trying to have some of those conversations, bring the public in, help them understand what we're trying to do, and come up with a plan that would be acceptable.
Thank you so much.
Yeah, thank you. Really appreciate that.
I think we've probably reached a point where we've exhausted our guest speaker. You've been very nice to us, and I really appreciate that. And I thank the public for very good discussions. Did you have any comments?
Really, no, I also appreciate your time and the commitment that you've shown towards reaching a good, solid, scientifically based report. But that feedback, I think you've asked for more feedback as well. And I guess my comment is to those of you in the audience to suggest for her some good locations where we could, in fact, have more of these sharing times to get that information going. So if you think about that as you're leaving, you might give her a slip of paper or something and say, here's an area group of people that would really respond. I'm thinking Grange Hall's being a natural gathering place, but schools and locations. So join in that conversation. Let's spread it throughout the region.
Yeah, adding that, because the presentation has been recorded, she doesn't have to listen to us, but I mean, the someone doesn't have to listen to us, but you can play that recording anyplace and focus on the presentation. I'm not saying you shouldn't go. I'm saying that I would be exhausted after a few of these.
Yeah, I think it's a combination. I appreciate that it is recorded and I can share it with, because I have had people ask, I can't make it. So I appreciate that it is recorded, but also recognize that you can learn the most by those personal interactions. So I appreciate that comment and I would love some suggestions. I'm relatively new to Washington, and so knowing who to reach out to and, not feeling like I'm imposing on people asking a favor. I have learned this trip that the county commissions are very receptive, and I appreciate that.
So just some housekeeping, just to remind people, the evening Wednesday session is every second Tuesday of the month for at least another 2 months.
And
At
the
which time
topics?
we'll reevaluate the effectiveness of having evening presentations. If anybody would like to contact the clerk of the board, how slippery I am, and let us know if this kind of a presentation, rather than a political presentation, more of a informative presentation, is something you would enjoy more. We can try and focus a little more and think about that. I'll just add a little hibbing here. My colleague hasn't said anything right now about that, so I'm still good. And we do not have any more comments, no executive session. I would like to call the meeting to a close. Are you good with that?
Sure.
This
I-
meeting's adjourned.
Thank you for coming.